you have downloaded the lecture note from the UFuture, right? Okay. So chapter 3, homeostasis. Okay. So these are the learning outcomes. In this chapter, we are going to look at homeostasis concept. Okay. Homeostasis concept. Describe the importance and mechanism of homeostasis. And then explain the negative feedback mechanism in controlling blood glucose level. In this part, we are going to look at the function of the hormone insulin and also glucagon in maintaining the normal set point of blood glucose level, and then explain homeostatic processes for thermoregulation involving form, function, and behavior. So this one, we are going to look at the adaptation of various types of animal in regulating body temperature. And then describe the structure and function of kidneys. So this one is the one that we looked at yesterday during lab. Okay, where you are required to know the structure of kidneys, the function of kidneys, osmoregulations, and how urine is formed. Okay, so therefore we are going to look at formation of urine, explain the role of the hormone ADH, or short form as antidiuretic hormones, and the hormone aldosterone in urine formation. And then lastly, explain the positive feedback mechanism, which is, for example, it involves childbirth. Okay, childbirth that involves contraction of uterus and as the result of the release of the hormone oxytocin. And also oxytocin causes the release of milk when the baby is breastfeeding. Okay. Okay, so because yesterday we for the lab we looked at the function of kidney and the urine formation. For this lecture, I'm going to skip the first part of the lecture note. We are going to look at the function of kidney. Okay, so this one we are going to look at on our next lecture. So we are going to go to this part of the lecture note, okay, which is osmoregulation of human. Okay. Okay, so homeostatic organ in this subtopic, osmoregulation of human. Okay. We are going to look at the homeostatic organ involved in osmoregulation, how urine is formed, and then the role of the hormone ADH and also aldosterone. Okay. So kidney, right? So kidney as the homeostatic organs. So the excretory organs of vertebrates function in both excretion and osmoregulation. So excrete nitrogenous waste in the form of urea. Osmoregulates by control solute concentration and balance, water gain and loss. First of all, you have to know the basic function of homeostasis. Homeostasis is the mechanism done by your body to maintain the normal physical and chemical condition of your body at the normal set point. For example, if your body is supposed to be, the temperature of your body is supposed to be maintained at 37 degrees. If you are in a cold environment, so your body will shiver, for example, to generate a heat to warm up the body. That is homeostatic. If homeostasis. Okay, so if you are eating too much rice, okay, so obviously in your blood the level of glucose will increase. So to lower back your blood glucose level to the normal set point, so that is the function of the hormone insulin produced by the pancreas, which is the excess glucose that you eat from the rice that is not used as the a source of energy. So the glucose will be converted into glycogen and stored in the liver. So this is done by the hormone insulin. Okay, so that is homeostasis, the mechanism done by your body to maintain the body normal physical and chemical factors at the normal set point. Okay, so for this part we are going to look at how your body maintain the right solute concentration and water balance gain and loss through osmoregulation, which involve the organ kidney. Okay, and also kidney function to remove wastes, okay, from your body fluid, especially nitrogenous waste in the form of urea. All right. So the mammalian kidney has two distinct regions, which is the outer part is the renal cortex, and the inner, which is the renal medulla. Okay, so in the kidney, waving back and forth across the renal cortex and medulla are the nephrons. Okay, so nephrons, it consists. Why does the statement says or describe the nephron as waving back and forth across the renal cortex and medulla? So the kidney has two regions, okay, which is the cortex, and this is the medulla. Okay, so you have the glomerulus, and then you have the Bowman capsule, and then from the Bowman capsule you have the proximal tubule, and then into the loop of Henle, okay, ascending loop of Henle, and then distal tubule, and then collecting ducts. Okay, so that's why it describes the nephron as waving back and forth across the renal cortex and medulla of the kidney. Okay, so the nephron function as the functional unit of the vertebrate's kidney. So the nephron is the structure within the kidney that function actually to filter the blood. Okay, so the two regions of the regions of the kidney, parts of the kidney. So the renal cortex. Okay, the renal cortex is the outer part of the kidney. It has a smooth texture. Is the location of the Bowman capsule and also the glomerulus, like I've drawn just before. Okay, so in addition to the proximal and distal tubule and the associated blood supplies. Okay, so this is the part of the renal cortex. The location of the glomerulus is actually blood vessels. Okay, and then you have the Bowman capsule, which is the tubule part of the nephron, and then this is the proximal tubule, and this is the distal tubule. So in the cortex region you have those components of the nephron. Okay, and then in the cortex, in the medulla, you have the collecting ducts, and also part of the you have the loop of Henle and also part of the collecting ducts. Okay, so you have to know the component of the nephrons. Okay, so and then the outer is cortex, the middle part or the inner part is the renal medulla. So that is the medulla, the inner part of the kidney. And then you have the renal pelvis. Okay, so it is a funnel shaped basin that receives the urine drained from the kidney nephron via the collecting ducts, and then to the larger papillary ducts. Okay, so this is the diagram to show you how the kidney looks like from inside. Okay, the cross section of kidney. So the outer part, okay, of the kidney, the most outer part, you have the capsule. Okay, the capsule, And then beneath the capsule you have the cortex. Okay, the cortex here, this region here is the cortex. Alright, and then you have the renal medulla, the lighter part here. Okay, and then you have the renal pelvis. So the nephrons function to filter the blood, and once the blood are filtered, it forms filtrate. Okay, it forms filtrate that will become the urine. So the filtrate that will become the urine will be collected into the renal pelvis here. So this is the yellow structure here is the renal pelvis. So the renal pelvis will collect the filtrate that is going to be the urine. Okay, and then the urine will be conveyed along the ureter and then into the urinary bladder. Okay, so okay, so you have the blood vessels. Okay, so you have the renal blood vessels, sorry, the renal artery, the red one here. Okay, the renal artery, blood flows into the artery, and then you have the renal vein, blood flows out of the kidney. Okay, out of the kidney. Renal artery, blood flows into the kidney. Renal vein flows out of the kidney. So, okay, so there are two types of nephrons. Okay, there are two types of nephrons. You have the juxtamedullary nephrons, and also you have the cortical nephrons. Okay, so what is the difference between juxtamedullary nephron and cortical nephron? It is based on the length of the loop of Henle. Okay, loop of Henle. So if you look at the juxtamedullary nephrons, okay, so here is the glomerulus of the nephrons, and then into the proximal tubule. Okay, and then from the proximal tubule into the loop of Henle. So the loop of Henle, going down you have the descending limb. Okay, going up you have the ascending limb of loop of Henle. So and then into the distal tubule and into the collecting ducts. Okay, so for juxtamedullary nephron, it has a longer loop of Henle. Okay, and as for the cortical nephrons, so this is the cortical nephron. So the glomerulus, the vessels, the blood vessels part of the nephron, and then the Bowman capsule, which is the tubule part of the nephron, and then into the proximal tubule, and into the descending limb of loop of Henle, ascending, and then into the distal tubule, and then into the collecting ducts. So for cortical nephron, it has a shorter loop of Henle. So what is the significance of having different length of loop of Henle? So therefore, for kidney, the function of the juxtamedullary nephron is for the animal or the organism adaptation to absorb more water. Okay, absorption of more water. Okay, more water. So here is where the descending limb of loop of Henle is where water will be reabsorbed. Okay, for animal that needs to conserve more water within the body, so they have more of the juxtamedullary nephrons. Okay, so to reabsorb more water from the body. So compared to the cortical nephrons, where the loop of Henle is much more shorter, so less water will be reabsorbed. Okay, so this is for animals that lives in hot area. They need to conserve more water. Okay, they need to reserve more water within the body, and for this animal, they will produce a more concentrated urine that are produced in low volume. Okay, low volume and much more concentrated. Okay, this is for animals adaptation, so that they can survive in a more hot area. Okay, so for mammalian excretory system, the mammalian excretory system centers on paired kidneys. So we have two kidneys. Okay, a pair of kidneys. So which are also the principal site for water balance and salt regulation. Okay, so water is reabsorbed, salts will be reabsorbed at some region of the kidney. Sorry, at some region of the nephron. So each kidney is supplied with blood by renal artery. Okay, renal artery, it allows blood to flow into the kidney, and then blood will be flowing out of the kidney by renal vein. Okay, so urine exit each kidney through a duct called the ureter. Okay, so from the kidney, before storing the urine into the urinary blood, into the urinary bladder. So it is the ureter. So ureter connects the kidney with the urinary bladder. Okay, so both ureters drain into a common urinary bladder, and urine is expelled out through the urethra. So you have to know the sequence, okay, of the flow of the urine, okay, from the kidney into the ureter, into the urinary bladder, and out of the body through the urethra. So you have to know the flow, okay, the sequence for the flow of urine. Okay, so this is the structure of the human urinary system. All right. Just wait a moment until the slide changes. I hope saya tak terkeluar lagi. Are you still on slide 44? Mammalian. Okay, it changes already. Right. So this is the diagram for the human urinary system. Okay. So our body has two kidneys. You have the right kidney here, the right kidney, and then you have the left kidney. Okay, the left kidney, and the red blood vessels is the renal artery. Okay, left renal artery. You have the right renal artery. Sorry, the blue one is the vein. Okay, right renal vein. So the blue is the vein. The red blood vessels is the artery. Okay. So, and then you have the inferior vena cava, the blue blood vessels, the larger blood vessels. Okay. And then you have the opening of the opening of the urethra. Okay. So the urine will be stored in the urinary bladder, and then out of the body through the urethra. Okay. So these the other large blood vessels, you have the abdominal aorta. Okay. The abdominal urethra. So the kidney filter the blood. Okay, the two kidneys filter the blood, and then the filtration of the blood produces filtrate in the nephron in the kidney, and then the filtrate will become urine. The urine will flow through the ureter, and then into the urinary bladder, and out of the body through the urethra. Okay, so you have the right, the right and the left ureters. Okay. Okay, so the structure of nephrons, alright. So the nephron consists of a single long tubule and a ball of capillaries called the glomerulus that filters the blood. Okay, so the nephron actually, it has two components: blood vessels and also tubules. Okay, blood vessels, it starts from the glomerulus. Okay, glomerulus. Tubule, it starts with the Bowman capsule. Okay, capsule. So the blood vessels, obviously, it carries blood. Okay, blood in the glomerulus that is covered by the Bowman capsule is where the process, we call it as ultrafiltration, occurs. Okay, so the high pressure in the glomerulus will force fluid from the blood to enter into the tubule part of the nephron, which is the Bowman capsule. So this fluid, once fluid enters into the Bowman capsule, we call it as filtrate. Okay, filtrate. Okay, so tubules from Bowman capsule into proximal tubule, into loop of Henle, into the distal tubule, and into the collecting ducts. Okay, so actually collecting ducts, it collects filtrate coming from several nephrons. Okay, coming from several nephrons. For the glomerulus, it receives blood from the afferent arteriole, and then from the afferent arteriole into glomerulus and out into the efferent arterioles. So surrounding the tubules, you have blood vessels surrounding the tubule. We call it as vasa recta. Okay, vasa recta are blood vessels surrounding the tubule part of the nephrons. Okay, so it says here, Bowman capsule surrounds and receives filtrate from the glomerulus. So the fluid that starts to enter into the Bowman capsule, we call it as filtrate. Okay, so if the fluid is inside the tubule, we call it as filtrate. Okay, so you have the Bowman capsule, the first region of the tubule. The second region of the tubule is the proximal convoluted tubule. And then the third part is the loop of Henle, and the next one is the distal convoluted tubule. Alright, so the function of the proximal tubule is for the reabsorption of water, salt, glucose, and amino acids. Okay, so so this is where the nutrients, okay, that gets filtered in the Bowman capsule will be reabsorbed into the from the proximal tubule. Okay, it will be reabsorbed back into your blood circulation. Okay, it will be reabsorbed back into your blood circulation and into your blood. So this will be reabsorbed back into your blood vessels. Okay, into the blood vessels, which is actually the vasa recta. So and then after the proximal tubule, you have the loop of Henle, which maintains the concentration gradient, okay, within the renal medulla, actually within the renal medulla region of the kidney. And then the distal convoluted tubule is the is where tubular secretion of hydrogen ions, potassium, and certain drugs occurs. So secretion, the term secretion is used for the movement of solute from blood, okay, from blood into interstitial fluid into the filtrate. Okay, filtrate, and it will become urine. Okay, that is the term secretion. For reabsorption is from filtrate into interstitial fluid into blood. Okay, in the blood vessels. That is reabsorption. Reabsorb back into your blood, into your body fluid, into your blood. Okay, so you have to know where to use the term. Okay, these terms. Okay, so this is the structure of nephrons. So as you can see, the outer region here, you have the cortex. Sorry, capsule. Okay, so this region here is the renal cortex, and this region here where you have the loop of Henle, okay, is the renal medulla. Okay, so you have to know the region of the kidneys. Okay, the regions of the kidney. So in the cortex region, you have the blood vessels part of the of the nephrons. Okay, nephrons. So this is the glomerulus, right? Okay, glomerulus. And then surrounding the glomerulus, you have the tubule part of the nephron. Tubule is where the filtrate is located. So this is the Bowman capsule. Okay, Bowman capsule, the tubule part of the nephron. And then you have the proximal tubule, and then you have the descending limb of loop of Henle. You have the ascending limb of loop of Henle, and then distal tubule, and then this is the collecting ducts. Okay, so collecting ducts is actually, if you refer to the reference book, the one that I read is Campbell. It says the collecting ducts, it collects filtrate coming from several nephrons. Okay, it collects. That's why you can see here you have several branches here. So these are the branches coming from several nephrons. Okay, that function to collect the filtrate that will become urine to be collected into the renal pelvis. Okay, so this is the detailed structure. Okay, so you have so for nephrons, you have to remember it has two components: the blood vessels part, and then the tubule part. Okay, the tubules part. Okay, so blood vessels, it starts with the glomerulus. The glomerulus will receive blood coming from the afferent arterioles. Okay, afferent arteriole receive blood coming from the renal artery. So from renal artery, blood flows into the kidney and into the afferent arterioles and into the glomerulus. And from the glomerulus, out of the glomerulus into the efferent arterioles. Okay, so the diameter, okay, between the afferent and efferent arterioles, the diameter of these two arterioles is different. Okay, the afferent arterioles, it has a bigger diameter compared to the efferent arteriole. It has a smaller diameter. Okay, if we were to look at the diagram in detail. Okay, so this is the afferent arteriole, this is the efferent arterioles. So the difference in the diameter causes a higher blood pressure. Okay, a higher blood pressure to occur. Okay, to occur in the glomerulus and causes the formation of filtrate in the Bowman capsule. Or is it vice versa? Sekejap. Saya kena make sure siapa yang besar, siapa yang kecil. Doesn't say. Okay. Nanti you all check lah siapa yang besar, siapa yang kecil. But basically, okay, the diameter between the afferent and efferent arteriole, it is different. Okay, one is smaller than the other one is bigger. So the difference in these two arterioles creates a high blood pressure in the glomerulus. Okay. So from the efferent arterioles, you have a net of blood capillaries surrounding the tubule part of the nephrons. Okay, so the blood vessels surrounding the loop of Henle, we call it as vasa recta. Okay, vasa recta. Okay, the blood capillaries surrounding the proximal tubule and the distal tubule, we call it as peritubular capillaries. Okay, so that is those are the component of blood vessels. So it starts with the glomerulus. Okay, and then you have the peritubular capillaries. Okay, peritubular capillaries are blood vessels surrounding the proximal tubule and distal tubule. Okay, and then you have the vasa recta. Vasa recta are the blood vessels surrounding the loop of Henle. Okay, so the tubule parts of the nephrons, it consists of the Bowman capsule, okay, proximal tubule, loop of Henle, and distal tubule. Okay, so these are the tubule part of nephron before it enters into the collecting ducts. Okay, collecting ducts. Okay, so you have to know the structure of nephrons, the components of the nephrons. Okay, so formation of urine. So nephron is organized for stepwise processing of blood filtrate. So you have the first process. Okay, the first process is filtration. What is filtration? It involves blood pressure, different in hydrostatic pressure and osmotic pressure, that causes this pressure causes body fluids to be filtered. Okay, so this actually occurs between the glomerulus and the Bowman capsule. Okay, Bowman capsule. Filtration occurs in that region, glomerulus and Bowman capsule. And then after that is reabsorption. So reabsorption occurs in the proximal tubule. It also occurs in the loop of Henle. It also occurs in the distal tubule. It also occurs in the collecting ducts. Okay, that is reabsorption. Reclaim valuable solutes. Okay, and then secretion, which is add toxin and other solute from the fluid, from your body fluid into the filtrate. So as I said before, for reabsorption, the term reabsorption is from filtrate into interstitial fluid into blood vessels. Okay, so the blood vessels, if it occurs in the cortex region, it involves the peritubular capillaries. If it occurs in the medullary part of the kidney, it involves the vasa recta. Okay, the blood vessels. For the secretion, it is from the blood. Okay, blood in blood vessels, sama ada glomerulus or peritubular capillaries or vasa recta, from the blood into interstitial fluid into filtrate. Okay, filtrate. So this filtrate later will become the urine. Alright. So kidney removes metabolic waste products such as urea, creatinine, and uric acid, and excreted them in the form of urine. Okay, the process of filtration, reabsorption, and secretion. Okay, so the first one is where filtration occurs, or we call it as the glomerular filtration. So for glomerular filtration, it involves this region, the Bowman capsule and also the glomerulus. Okay, and then after filtration, due to the high pressure, due to the high hydrostatic pressure in the blood, that causes fluid from the blood to be filtered and enters into the Bowman capsule. And then in the Bowman capsule, you have the formation of filtrate. Okay, you have the formation of filtrate. So the filtrate that enters into the Bowman capsule then flows into the proximal tubule. So in the proximal tubule is where tubular reabsorption occurs and also tubular secretion occurs. Okay, so and then after the proximal tubule, you have the loop of Henle. Okay, so loop of Henle also occurs reabsorption. Okay, reabsorption occurs in the loop of Henle, and then you have tubular reabsorption and secretion that also occurs in the distal tubule. Okay, so proximal tubule, distal tubule, it occurs reabsorption and secretion. Okay, distal tubule also occurs reabsorption and secretion. Alright, and then loop of Henle also it mostly involves reabsorption of water. Okay, so it involves reabsorption of water in the descending limb of Henle, and ascending it reabsorbed salt, NaCl. Okay, it reabsorb NaCl in the ascending limb of loop of Henle. And then from the distal tubule, it goes into the collecting ducts. In the collecting ducts, it occurs reabsorption of water again. Okay, so the body, okay, the body needs to reabsorb or reserve as much water as possible. Okay, so before the urine is formed. Okay, so if your body wants to conserve water, so the urine form will be concentrated, and the volume of the urine will be low. Okay. Okay, so the first process that occurs is the filtration. Okay, filtration of blood. So the process occurs in the glomerulus. We call it as glomerular ultrafiltration. Okay, So it says here Bowman capsule envelopes, okay, it surrounds the glomerulus. Urine formation begins here. Okay, so blood flows through the glomerular capillary under high pressure, forcing more than 10% of the plasma out of the capillary into the Bowman capsule. Okay, so filtration occurs as higher blood pressure or hydrostatic pressure forces fluid from the blood into the glomerulus, into the lumen of Bowman capsule. Okay, so tadi apa? You have the afferent arteriole flows blood into the glomerulus, okay, into the blood vessels, okay, in the glomerulus and out into the efferent arterioles. These two arterioles will have different diameter. You go back and check which one will have the higher, which one will have the bigger diameter. Okay, so this difference in the diameter causes a high blood pressure in the glomerulus, and the blood in the glomerulus will be filtered into the Bowman capsule surrounding the glomerulus. Okay, so to this point, because 10% of the blood, 10% of the plasma in the blood is filtered into the Bowman capsule, so it means that the plasma contains the valuable stuff as well in the filtrate. Okay, it has water, it has amino acid, it has glucose, it has other nutrients. So these valuable stuff need to be reabsorbed back. Okay, reabsorbed back into your blood circulation, which is the next process that occurs after filtration. Okay, so it says here factors contribute to the production of glomerular filtrate. Okay, glomerular filtrate are the fluid in the Bowman capsule. Okay, so the first point. Okay, so these are the factors that contribute to the production of glomerular filtrate, which is the first one. Hydrostatic blood pressure in the glomerular capillaries is higher than in other capillaries. So higher pressure due to the high resistance to outflow presented by the efferent arteriole, which is smaller in diameter than the afferent. Says here. Betul lah saya cakap tadi kan. So afferent arterioles will have the bigger diameter compared to the efferents. So the difference in diameter causes the blood to flow in high pressure in the glomerulus. Okay. So the second factors that contribute to the formation of glomerular filtrate is large surface area. Okay, large surface area for filtration provided by the highly coiled glomerular capillaries. So if you refer to the structure of the nephron earlier, you will find the glomerular, it contains a dense or a coiled, a ball of capillaries. Okay, something like that. So in the glomerulus, it has a higher density of blood capillaries in the glomerulus. This is to create a higher surface area. So more blood can be filtered to form the filtrate. Okay, more blood can be filtered to form the filtrate in the Bowman capsule. And then the third factor is great permeability of glomerular capillaries due to numerous small pores between the endothelial cells make the glomerular capillaries more porous than typical capillary. So the glomerular capillaries, they have more pores, they are more porous. So this allows the formation of more filtrate in the Bowman capsule. Okay, so it is more permeable. Glomerular capillary is more porous. Okay, because it has more pores between the endothelial cells. Endothelial cells are the cells surrounding these structures, surrounding the glomerulus. Okay, so it says here blood is carried by afferent arterioles. So afferent arteriole, it conveys blood coming from renal artery. Okay, so you have the renal artery, and then blood flows into the afferent arterioles into the glomerulus and out into the efferent arterioles. Okay, Okay, so blood is carried by an afferent arterioles into the glomerulus. The blood is filtered as the blood pressure forces fluid through the porous capillaries, walls, and filtration slits. Okay, so these are the features of the wall of the glomerulus and also Bowman capsule. They have pores and filtration slits. Okay, so glomerular capillaries permeable to water and small dissolved molecule, for example salt, glucose, amino acid, vitamins, urea, and nitrogenous wastes. Okay, so from these solutes that enters into the Bowman capsule as filtrate, these substances needs to be reabsorbed back into your blood. Okay, so the next process that occurs after filtration is reabsorption. Okay, so but this one will be maintained. Nitrogenous waste, urea will maintain in the filtrate. Right, so glomerular capillaries not permeable to blood cells or large molecules such as plasma protein. Okay, so for larger molecules, blood cells or plasma protein, they will not be filtered. They will not be part of the filtrate in the Bowman capsule. Okay, so blood moved out into the efferent arteriole. So for example, if your urine contains protein, so it means that there is problem in your kidney. Okay, you have disease related to the kidneys because the protein supposed to be maintained. Okay, it is supposed to be maintained in the blood, not enter into the Bowman capsule to become part of the filtrate. So if you have blood in your urine, if you have protein in your urine, so it means that there's something wrong with the kidney. Okay. Okay, so cutaway view of the Bowman capsule. So as you can see here, so these are the region that is involved in ultrafiltration. Okay, ultrafiltration or filtration. You have the glomerulus, and surrounding the glomerulus you have the Bowman capsule. Okay, so this is for the so this is it is the same for any nephron, juxta or cortical nephrons. Okay, sama je. And then the feature of the glomerulus, it has podocytes. Okay, podocytes, the cell of the glomerulus. So the cell of the glomerulus, the wall of the glomerulus, they have pores, they have small pores. Okay, and also filtration slits. So this allows blood from the glomerulus to enter into the Bowman capsule to become filtrate. Okay, so you have the glomerular capillaries, you have the afferent arterioles. Afferent arterioles will have bigger diameter compared to the efferent arterioles. Efferent is the one that allows blood to flow out. Okay, if afferent into efferent out of the glomerulus. Okay, so these are the structures that involve in ultrafiltration. Okay, so podocytes. Okay, the features of the glomerulus and Bowman capsule. All right. So if you were to go into detail to the structure of the glomerulus and the Bowman capsule. Okay, so as you can see here, so this is the wall of the glomerulus. So as you can see, it has many pores. Okay, so in the glomerulus is where blood will flow. So in the glomerulus you have blood, you have blood cells. Okay, red blood cells. So if we were to look in detail into the wall of the glomerulus, as you can see, it has many pores. Okay, so the wall of the blood vessels or of the glomerulus, okay, is made up of cells. We call it as endothelial cells. Okay, endothelial cells. Cells lining the glomerulus, we call it as endothelial cells, and they are very porous. Okay, so podocytes are cells in the wall of Bowman capsule, uh, contact with capillaries of the glomerulus. So podocyte ni, okay, so podocytes are the cells, okay, are the cells of Bowman capsule. Okay, podocytes are cells of the Bowman capsule which is in contact with the glomerulus. And then you have the filtration slits. Okay, process of adjacent podocytes are separated by narrow gaps. Okay, narrow gaps, that is filtration slit yang ni. Okay, filtration slits. Okay, narrow gaps that allows fluid plasma from the glomerulus to enter into the Bowman capsule to become the filtrate. So it says here filtration membrane of kidney. So the porous wall of the glomerular kidney and the filtration slits, okay, filtration slit, the structure of the Bowman capsule that has gaps, okay, between the podocytes form a filtration membrane. The membrane is highly permeable to water, small molecules and ion, but restricts the passage of blood cells and large molecules. Plasma protein, blood cells cannot be filtered out, okay, cannot be part of the filtrate in the Bowman capsule, okay, due to the bigger size. So this one you have to remember, okay, the structure, the feature of the glomerulus and also Bowman capsule. Okay, so pathway of the filtrate. So after the Bowman capsule, from the Bowman capsule, the filtrate passes through the region of proximal tubule. So you have to know the sequence, the flow of the filtrate. So from the Bowman capsule into proximal tubule, into loop of Henle. Loop of Henle has two regions, descending and ascending. And then from ascending into distal tubule, and from distal tubule into collecting ducts. So these are the flow of filtrate. Okay, so these are the tubule. Okay, the tubules part of nephron. Okay. Okay, so from this diagram, it again shows you the structure of nephrons. Okay, so you have to know the structure of nephrons. You have to know the labeling of the nephrons. Okay. So as I said, nephron, it has two components, blood vessels and tubules. Blood vessels for the flow of blood, tubules for the flow of filtrate that will become urine later. Okay, so the red blood vessels is carrying oxygenated blood. Okay, the blue blood vessels, it carries deoxygenated blood. So blood enters into the kidney that carries nutrient, that carries oxygen. Okay. So it means that from renal artery, okay, from renal artery into afferent arterioles, into the glomerulus, and out from the glomerulus into efferent arterioles. Okay, so from efferent arterioles, okay, from efferent arterioles into peritubular capillaries. Peritubular capillaries, it surrounds proximal tubule and it also surrounds distal tubules. Okay, proximal tubule, distal tubule is surrounded by peritubular capillaries, and then from proximal tubule it goes down into descending limb of loop of Henle. Sorry, okay, proximal tubule, then going down you have the descending limb of loop of Henle. Okay, descending limb of loop of Henle, and then going up, okay, is ascending limb of loop of Henle that enters into the distal tubule, enters into the collecting ducts. Surrounding the loop of Henle, you have the blood vessels. We call it as vasa recta. Okay, so if you observe here, the flow of blood, okay, in the blood vessels in the vasa recta, which is surrounding the loop of Henle, the flow of blood will be in the opposite direction, okay, in the opposite direction as the filtrate in the loop of Henle. So if you notice here, okay, okay, so the blood in the vasa recta will flow in opposite direction as the filtrate in the loop of Henle. So they will flow in opposite direction. So this is very significant when we looked at the detailed process of how urine is formed later. Okay, the flow of fluid in opposite direction, we call it as counter current. Okay, in opposite direction. Blood flows in opposite direction as filtrate, we call it as counter current. Okay, so counter current exchange. When the blood and the filtrate flows in opposite direction, they will exchange. Okay, they will exchange substance or molecules. Okay, they will exchange water and also salt. Okay, so later we are going to look at from which direction. Okay, so the movement of these two molecules. Okay, so this is the structure of nephron that you have to know. Okay. Okay, so proximal convoluted tubule. Okay, proximal convoluted tubule after the Bowman. It is after the Bowman capsule. So it is the longest and widest part of the nephron. Okay, so molecules are transported actively and passively from the filtrate in the Bowman capsule. Okay, in the Bowman capsule into the peritubular capillaries. So remember the previous diagram. Peritubular capillaries are the capillaries surrounding the proximal tubule and also distal tubule. Okay, so in this region, okay, salt, which is NaCl in the filtrate in the Bowman capsule, enters the cell of the transport epithelium by facilitated diffusion and also co-transport mechanism. What is transport epithelium? Transport epithelium is the name of the cell, okay, is the name of the cell that surrounds or that becomes the wall of the tubule. Okay, the wall of the tubules. Okay, so maksudnya cell surrounding the proximal tubule, sel yang jadi wall of proximal tubule, sel yang jadi wall of loop of Henle, okay, sel yang jadi wall of distal tubule, sel tu they are one cell thick and they are called as transport epithelium. Okay, so the tubule is surrounded by wall and the wall is one cell thick and it is called as transport epithelium. So this is the lumen of the tubule. So for example, this is the lumen of the proximal tubule that contains the filtrate. Okay, so substance such as, for example, water, substance such as amino acid in the filtrate will enter into this cell, into the transport epithelium, either through the process of facilitated diffusion, okay, from high concentration. Okay, facilitated diffusion is passive, kan? Passive from high concentration to low concentration, from filtrate back into the interstitial fluid into the blood vessels. Okay, blood vessels in this case is peritubular capillaries. Okay, so it depends on the type of molecule. Okay, so if the molecule requires facilitated diffusion, so it will travel through the transport epithelium through facilitated diffusion. If the movement of the solute requires transport protein, then it is through co-transport. Okay, co-transport. So both basically requires transport protein lah, kan? Transport protein, because they both because these two movement, okay, requires transport protein to move solute. In this case is sodium chloride. Okay, sodium chloride. And then as salt moves from the filtrate into the interstitial fluid, water flows by osmosis and thus reducing filtrate volume. Okay, filtrate volume. So water will also be reabsorbed. Water from the filtrate in the proximal tubule will also enter into transport epithelium, enters into the interstitial fluid and enters into the blood capillaries, which is the peritubular capillaries. when water is reabsorbed, so therefore the filtrate volume will reduce. Okay, so and then the next substance that will be reabsorbed is glucose, the nutrients, okay, which are glucose, amino acids, potassium ion, and other essential substances are also actively or passively transported from the filtrate into the interstitial fluid, then into the peritubular capillaries. Okay, so this is the process of reabsorption that occurs in the proximal tubule. And it says here some toxin materials such as drugs and also toxin processed from the liver are secreted into the filtrate by active transport. Secreted, pergerakan dia daripada mana? From blood, okay, from blood into interstitial fluid into the tubule, in this case into the proximal tubule, that is secreted. So toxin from the blood, drugs, okay, that are filtered in the liver will enter into the kidney and will be secreted into the into the into the proximal tubule, okay, to the proximal tubule to become part of the filtrate. It become part of the filtrate in the proximal tubule. Okay, so you have to know which are the substance that will be reabsorbed. You have to know which are the substance that will be secreted into the filtrate. Okay, so okay, so if you look at the next slide, so processing filtrate in this region helps to maintain a relatively constant pH in the body fluid. Cells of the transport epithelium, as I said before, transport epithelium is the name of the cells surrounding, or it makes the wall of the tubule. Okay, that is transport epithelium. So cells of the transport epithelium secretes hydrogen ion into the lumen of the tubule. Okay, so secretes daripada mana? Blood into interstitial fluid into the tubule. Okay, to become part of the filtrate. Okay, to become part of the filtrate. So hydrogen ion are secreted into the lumen of the tubule, synthesize and secrete ammonia, act as a buffer to trap hydrogen ion in the form of ammonium ion, NH4+. That is the ability of the cell of the transport epithelium. It synthesizes and secrete ammonium, okay, to function as buffer to trap hydrogen ion from the blood. So this is to maintain the normal blood pH. Okay, the normal blood pH has to be maintained at 7.4 if I'm not mistaken. Okay, 7.4. Kita tengok awal-awal slide. Nah, 7.4. The normal blood pH has to be maintained at 7.4. So that is the significance, another significance of filtration of blood that occurs in the body. Okay, which is to maintain the normal pH of blood. Okay, so that is the function of the transport epithelium, to synthesize substance that will become buffer, that will bind with the hydrogen ion from the blood to and form ammonium ion. Okay, ammonium ion. Okay, so transport epithelium, which is the cells surrounding the proximal tubule, will synthesize and secrete ammonia that will bind hydrogen ion from blood. Okay, so if your blood has high concentration of hydrogen ion, or if your blood is acidic, this excess hydrogen ion will enter into the proximal tubule through the process of secretion. So this hydrogen ion in the tubule, okay, in the filtrate, hydrogen ion, okay, hydrogen ion from blood, okay, from blood enters into the filtrate. Okay, it enters into the filtrate. It binds with ammonia, okay, and NH3, Okay, so NH3 plus H plus, so it becomes ammonium ion. Okay, ammonium ion. So this is to make sure your blood pH is maintained at the normal set point. One of the process. Okay, one of the mechanism to maintain the normal blood pH. One of the process. Okay. So the more acidic the filtrate is, the more ammonia the cell produces and secretes. Okay, so that is the ability of transport epithelium, synthesizing and secreting ammonia into filtrate so that the ammonia can bind with the hydrogen ion that enters into the filtrate through the process of secretion. So this is to lowers the blood pH back to the normal set point, which is 7.4 if your blood is acidic. Okay. So the tubule also reabsorb about 90% of the buffer bicarbonate, which is this ion, bicarbonate ion, HCO3 minus, okay, from the filtrate. Okay. So, okay, so this ion, bicarbonate ion, okay, which end up in the filtrate, will be reabsorbed. Okay, will be reabsorbed and back into the blood. Okay, why does your body needs to reabsorb back the bicarbonate ion? Because the bicarbonate ion will be used as one of the ions that will also maintain the normal pH, the normal blood pH. Okay, the normal blood pH. It involves in carbon dioxide transportation later that we are going to look at in chapter respiration. Okay, so it relates with the carbon dioxide transportation in the blood. Okay, this bicarbonate ion. All right. So the filtrate volume decreases, materials to be excreted become concentrated, because in the proximal tubule it also occurs reabsorption of water reabsorbed, okay, and causes the filtrate volume to decrease, and also the reabsorption of the nutrients. Okay, so this decreases the filtrate volume that will enter into the next region, which is a loop of Henle. Okay, it says here materials to be excreted become concentrated. So from proximal tubule into the next region, which is the loop of Henle. Okay, so by looking at this diagram, as you can see, so this is the process that occurs in the proximal tubule. Okay, in the proximal tubule. So proximal tubule is in the cortex region. Okay, it is in the cortex region of the kidney. Okay, the cortex region. So in the cortex region, you have the glomerulus, you have the Bowman capsule that involve the process of ultrafiltration, and the filtrate that enters into the proximal tubule. It has water, it has other valuable stuff, valuable nutrients that needs to be reabsorbed. Okay, so the filtrate, it contains water, it contains salts, it contains bicarbonate ion, hydrogen ion, urea, glucose, and also some drugs. Okay, so water will be reabsorbed, salt will be reabsorbed, bicarbonate ion will be reabsorbed, hydrogen ion will be secreted, urea will be secreted, okay, and glucose, amino acid will be reabsorbed, some drug will be secreted. Okay, secreted means into the filtrate, reabsorb, it means that from filtrate into blood. Okay, from filtrate into blood, from filtrate into blood. Okay,